So today's lecture is on the revision of protein metabolism. As we know about the basics of protein, the proteins are defined as the polymers of L-alpha amino acids, and they are joined by the peptide bonds. They are building blocks in various cells, help in the formation of the various tissues. Classification is based on the 4 different types, depending on their nutritional requirement, depending on the amino acid content, how many essentials are present, how many essentials are absent. Like this, there are 4 types of classification. Amino acids, as you know, monomeric form, they are joined by peptide bond. The classification of amino acid, again depending on the side chain every amino acid has, it may be acidic, it may be basic, it may have sulfur containing side chain, or it may have the aromatic side chain. The nutritional requirement, on the basis of nutritional requirement, the amino acids can be classified as essential, non-essential amino acids. The structure of proteins, the proteins can be organized into primary, secondary, tertiary, quaternary structures that you have seen, must know from the protein chemistry. In the secondary structure, there is alpha helical, beta plated sheet. Tertiary structure is a 3D structure, and quaternary structure is the combination of 2 or more tertiary units, tertiary proteins, to form the quaternary structure. Then there were 2 terms, denaturation and coagulation. The denaturation is nothing but the disruption or the breaking of secondary, tertiary and quaternary structure to the primary structure. So only peptide bond remains, rest bonds break, and the protein is said to be denatured protein. Opposite, and the denaturation can be converted to its native form. That means if denaturing agent is removed, the primary structure, it can be converted back to its native structure, maybe quaternary, tertiary or secondary. So denaturation can be renatured. And the difference between denaturation and coagulation is the coagulation cannot be, or the coagulated protein cannot be renatured, it cannot be converted to its native form. So it is permanent denaturation. Then the plasma proteins, in the last part of protein chemistry, we must have studied the plasma proteins, which are divided into 3 parts. The most abundant is albumin, followed by globulins, and finally fibrinogens with their respective functions. So these are the points we must know from the chemistry of proteins. So these serves as a base to understand the protein metabolism. So these are the various objectives of the first lecture. The sources of proteins, the first class proteins. The proteins, they are called first class when all 10 essential amino acids are present in the protein. As you know, the amino acid nutritional, as per the nutritional classification, they are classified into 2 classes: the essential amino acids and non-essential amino acids. Essential are those which are need to be taken through diet daily, because they are not synthesized in the body. So if all the essential amino acids are present in the protein, they are called as the first class proteins. The examples are milk and milk product, eggs, the albumin of the egg, it has all essential amino acids, also fish and meat. So mainly the dairy and the non-vegetarian products are the sources of first class proteins. Then the second is second class proteins. These proteins are deficient in 1 or 2 essential amino acids. Out of 10, 1 or 2 essential amino acids are absent. Examples are rice, wheat and pulses. Now coming to digestion of proteins, in the protein digestion there are various types of enzymes that are utilized to digest the proteins to their simpler form, and those proteins are referred as proteolytic enzyme. And most of the proteins are digested or converted to their simpler form or smaller form due to cooking and by the action of the gastric axial in the stomach. And like lipid, again the proteins are not digested in the mouth, their digestion starts in the stomach. So So the enzyme acting on the proteins, they are called as proteolytic enzymes. They are responsible for breakdown of proteins. They are secreted by stomach, pancreas and small intestine. They are converted or they are subclassified into two types, endopeptidases and exopeptidases. Endopeptidases are those which act on internal peptide bonds of the polypeptide chain. I will show you the figure. So as you can see in this figure, this is the polypeptide chain, and all the peptide enzymes are classified either into endopeptidases or the exopeptidases. The endopeptidases, they can act on the peptide bond in the middle of the protein, not at the end. As we know, there is always N-terminal and C-terminal. These N-terminal and C-terminal amino acids, the bond between, the peptide bond between the first and the second amino acid at each terminal is broken by exopeptidases, and the rest, the middle peptide bonds are broken by endopeptidases. Got it? So endopeptidases break the peptide bond in the middle of the protein peptide chain. Exopeptidases are further subclassified into two classes. Aminopeptidases, that separates the amino acid, the end separate amino acid, which is present at the amino terminal. And second exopeptidases are the carboxypeptidases. They separate the amino acid which is present at the carboxy terminal or C terminal. So these are the two main classes, endopeptidases and exopeptidases. Exopeptidases are further classified into carboxypeptidases and aminopeptidases. Apart from this, there are other peptidases, dipeptidases, because by the action of endopeptidase and aminopeptidase, at the end there will remain only two amino acids, which has one of which has the amino terminal, second of which has a carboxy terminal. So neither of these, endo and exopeptidase, will act on this dipeptide. Instead, the dipeptidase will act and break the only peptide bond present in that dipeptide and convert it to two separate amino acids. So digestion of proteins, there is no digestion in the mouth. The proteins which are cooked, which are semi-digested by cooking, they are transported to the next organ, that is stomach. And in the stomach, the action of HCl, the hydrochloric acid secreted by the gastric glands, activate one of the proteolytic enzymes secreted by stomach, pepsin. It is first secreted as pepsinogen, converted to pepsin. So pepsin will act on the proteins and convert it to the smaller, the larger proteins are converted to smaller proteins by the action of the enzyme pepsin as well as the hydrochloric acid. Next, the semi-digested proteins then travel to small intestine, and in the small intestine, the proteases, the enzyme proteases, which are secreted by pancreas, they convert the semi-digested proteins to small peptides, and some free amino acids are liberated. So these proteases are secreted by pancreas. Now these small peptides and amino acids are converted to their simplest form. The proteins are converted to their simplest form, which is the requirement for the next step, that is the absorption. So they are soluble in aqueous medium. They can be easily transported to intestinal lining, intestinal epithelial cells, where active transport happens and the amino acids are absorbed into the blood, and once absorbed they are transported to the site of requirement where they are required. So dietary proteins, nearly about 100 grams per day we take through diet. They are not digested in the mouth, rather they are transported to stomach, and in the stomach by the action of the HCl, hydrochloric acid. HCl is one of the denaturing agents, so denaturation is caused by HCl of these proteins. So the proteins unfold, and one of the endogenous proteins, nearly about 100 grams per day, are also degraded. They undergo metabolism. So the enzyme pepsin secreted by stomach acts on the denatured protein. So they are converted to their simplest form, smaller proteins. Then they travel to the small intestine, where the pH is little higher than the stomach, and the enzymes that are secreted by pancreas are poured into the small intestine. The enzymes include trypsin, chymotrypsin, elastase, carboxypeptidase. These proteolytic enzymes act on the protein and convert them to small peptides or the separated amino acids. These further travel to the intestinal lumen, and in the intestinal lumen, via these intestinal cells, they are absorbed by the active transport. I hope you can see the slides changing, right? Can anybody answer? Okay. Okay, thank you. So the zymogens, as I told you, the pepsin, the enzyme, it is not synthesized as pepsin. It is first synthesized as its proenzyme or the zymogen form, which is the inactive form. Can anybody tell me why this is required, the zymogen or proenzyme form? Why pepsin or trypsin or chymotrypsin are not synthesized in their active form directly? Any idea? Yes, autodigestion, to inhibit the autodigestion of the tissue itself, because pepsin is synthesized from the gastric gland as pepsinogen. If it is synthesized as pepsin in the absence of food, that pepsin start digesting the stomach cells or stomach proteins. If chymotrypsin, it is synthesized as trypsinogen first, zymogen form. If it is synthesized as trypsin itself in the pancreas, then it will start digesting the proteins present in the pancreas, so it will cause damage to that organ. That's why the zymogen forms are synthesized first, and in the presence of food, in the presence of protein, these zymogen forms are converted to their active form. As I said, if the enzymes secreted by pancreas are activated without entering the small intestine, they are secreted in active form itself in the pancreas. The pancreatic cells will undergo damage or the digestion that will lead to the damage to pancreas, the whole pancreas, causing the condition called as acute pancreatitis, and it is life-threatening. So pepsin, it is a proteolytic enzyme that acts at pH. So these are basics. I hope I'll share these PowerPoints. You can go through if you don't understood. If not understood, let me know. We'll discuss it. Then there is one enzyme called renin, double N-I-N. There is one more hormone called renin. It is present in the kidney, excretory system, in the renin-angiotensin system. These two are different. Renin is an enzyme, and this double N wala renin is enzyme, and single N wala renin is an hormone. This double N renin, this renin is useful for the digestion or the curdling of the milk, and it is present in the infant. It is only present in the infants, not in the adults, because in the infants the exclusive source of the food or the energy is the milk from the mother, and that milk, to undergo digestion of the proteins and absorption, first it has to be curdled. So the proteins will be available for the action of these proteolytic enzymes, and they are digested easily due to this curdling of the milk. And for this curdling, this enzyme renin is important. It converts the abundant protein casein present in the milk to paracasein, and by further addition of calcium, it causes formation of calcium paracasein that forms the curdle or the coagulums of the milk, which increases the surface area for the proteolytic enzymes. Once the amino acids are formed, the amino acid, by the active transport, they are absorbed in the intestinal cell, and from the intestinal cell they are transported to the portal vein. As you can see in the figure, the amino acid, once formed in the intestinal lumen, it is co-transported along with sodium. There is co-transport of sodium along with the amino acid with the help of the protein transporter. Once the amino acid is inside the intestinal cell, for its transport from the intestinal cell to the blood, it requires 1 ATP, and it is one amino acid is transported into blood in the exchange or when the sodium is transported outside of the epithelial cell in the exchange of potassium. So this exchange of sodium potassium happens with the help of sodium potassium ATPase enzyme, which requires energy. So one ATP is utilized and converted to ADP and inorganic phosphate. That's why the transport of amino acid from the intestinal lumen to portal vein is called as active transport, because amino acid when enters the cell along with sodium, if it is to be entered in the blood, this amino acid there should be exchange of sodium with the potassium, and this requires ATP, and during this exchange the amino acid is pumped out into the portal vein via some transporter, the facilitated transporter. That's why the active transport. So it requires ATP for its transport from intestine to blood. So there are 5 different carrier proteins for different amino acids. As we have seen these in the transport, these are the carrier proteins. These carrier proteins are different for different amino acids, but collectively there are 5 carrier proteins which transport a set of amino acids. One is for neutral amino acids like alanine, methionine, valine, phenylalanine, tyrosine. One is for basic amino acids that include lysine, arginine and cysteine. One is for amino acid, that is proline and glycine. Acidic amino acids have different carrier protein that include aspartate and glutamate. Beta amino acids, which has the beta, which is a beta alanine, which has the imidazoline. So it has the separate carrier proteins. So the next is gamma glutamyl cycle. One way of transport of sodium from intestinal lumen to the portal vein is the active transport. The second way is gamma glutamyl cycle, or it is also called as a Mister cycle. It is a minor way of absorption of amino acid. It is present only in 2 organs, that is kidney and brain. It is energetically expensive because the active transport requires only 1 ATP, while the gamma GTT or GTT transport mechanism of amino acid requires 3 ATPs for 1 amino acid. And in comparison to production of amino acid, the utilization of production of energy from amino acid, the utilization of ATP is much more higher. So it is the secondary or very less utilized transport mechanism for the amino acid. It uses the protein glutathione, which has 3 amino acids, the glycine, cysteine and glutamate, and it acts as a transport protein in this cycle. It is useful when rapid transport of amino acid is needed. The intact proteins can never be absorbed in the blood. If they are absorbed, they show some immunological reaction. That is what we call the allergic reaction in adults. But in infants, the intact proteins can be absorbed, because as we know, the colostrum, the first milk from the mother, or some immunoglobulins are already present in the mother's milk that provide the immunity to the fetus or to the infant. Those immunoglobulins can be directly, intactly absorbed in the intestinal cell and then into blood. Those immunoglobulins provide the immunity to the infant. That is the only exception, but in the adults, no protein is directly or intactly absorbed. Okay? If absorbed, that shows the allergic reactions. There is one more exception, that is the vaccines. In some cases, the proteins are covered in some molecule, lipid molecule, and they can be intactly absorbed. The vaccines are the exceptions, but not endogenously protein. That the proteins that we take through diet cannot be absorbed in the intestinal cells. Then it causes the food allergy that we have seen. Hartnup disease, the defect in the intestinal and renal transport and absorption of neutral amino acids, including tryptophan, it causes the Hartnup disease. There are various transport proteins, the 5 transport proteins as I showed you. Most of the proteins, the neutral amino acids are transported with the specific transporter. If defect in that transporter, it causes the Hartnup disease. So there is one concept during lipid metabolism. I told you the fatty acid pool was there. It is not as such the fatty acid is present in some compartment in the body. Wherever the free fatty acid is present, it constitutes a fatty acid pool. Similar way, the amino acid pool. Even this point also we discussed during the lipid metabolism, the lipoprotein wala. So the amino acids, any free amino acid present in the body, wherever it is present, it constitutes the amino acid pool, free amino acids. There is no such compartment that at this place the amino acids are collected and stored. It is everywhere. Any free amino acid constitutes or contribute to amino acid pool. From these amino acid pool, the proteins can be synthesized. Or if the lifespan of a certain protein, for example hemoglobin, is over, these proteins can be degraded to amino acids or amino acid pool. The second source for amino acid pool is the dietary proteins. We eat the proteins through diet. Those undergo catabolism to simpler form, that is amino acids, and these amino acids by absorption they contribute to this amino acid pool. The next is synthesis of non-essential amino acids. There are 10 non-essential amino acids, right? So these non-essential amino acids may not be present in the diet. Diet may only have the essential amino acids, but these non-essential amino acids can be synthesized from essential amino acids, and once synthesized, they also can add to amino acid pool. Now, what are the fates of the amino acid present in the amino acid pool? One is the synthesis of tissue non-protein nitrogenous substances. Non-protein nitrogenous substances. So the proteins like heme, glutathione, several hormones, because we know all the hormones are proteins, all the enzymes are protein. The melanin pigment, the skin color pigment, the purines, pyrimidines, as we have seen, glycine is required for the synthesis of purines. The amino acid aspartate is required for the pyrimidine synthesis. The amino acid glutamate in the form of glutamine is required for as an amino group donor in both purine and pyrimidine synthesis. The protein creatine is synthesized from the glycine, arginine, these two amino acids, which is present. This creatine is present in the muscles, which is helpful for the storage of energy in the form of creatine phosphate. And during catabolism of amino acid, the amino group is liberated as ammonia, which is converted or detoxified to urea by the way of urea cycle, and the fate of this urea is to get excreted in the urine. Okay. The other fate is, once the amino acids undergo catabolism, they liberate via the initial reaction, transamination and deamination. The amino group is separated, liberated as ammonia, it undergoes urea cycle, and through its fate it is excreted as urea. What happens to the remaining carbon skeleton? The carbon structures from the amino acid, they are called as alpha keto acids. These alpha keto acids has various fates. If the amino acid is glucogenic, the alpha keto acid carbon skeleton can be converted to glucose. If it is ketogenic, it can be converted to acetyl CoA, it can be converted to some fat. Synthesis of non-essential amino acids, the carbon skeleton of these amino acids can form or generate non-essential amino acids, or it may undergo the formation of acetyl CoA. For example, one of the keto acid formed is pyruvate. The pyruvate may undergo further conversion to acetyl-CoA, and that acetyl-CoA then enter the PCA cycle and completely oxidize to carbon dioxide and water molecule and generate the ATP, the energy. So this is what the contributors of the amino acid pool and the fate of amino acid from the amino acid pool. The protein turnover, the breakdown of the protein in our body is almost equivalent to generation of the new protein. So synthesis is equal to breakdown, and this cycle is called as, or the balance is called as protein turnover. In any given adult, the protein synthesis is almost equal to protein breakdown. So protein turnover is protein synthesis almost equal to protein breakdown. So the condition is called as equilibrium. So hypertrophy, similar, the protein synthesis goes on. Once the lifespan of the protein is over, it undergoes breakdown, adds to amino acid pool. From the amino acid pool, again the cycle goes on, synthesis, breakdown, synthesis, breakdown. So synthesis is almost equivalent to breakdown, that is the equilibrium. Hypertrophy occurs when there is more synthesis. The muscle or the cells become more rigid due to excessive protein than the breakdown. So the next part is amino acid metabolism with respect to fate of amino group and fate of carbon skeleton, that is urea cycle. We are going to study and describe the mechanism, process of transamination and deamination. Then what happens to ammonia, what is the fate of ammonia, the urea cycle, and the functions of glycine. So fates of amino acids, body protein synthesis, as we have seen, peptide formation like glutathione, oxytocin, synthesis of non-protein nitrogenous substances like urea, creatinine, ammonia, uric acid, they are formed. Then amino acid, the major fate is they may undergo transamination followed by deamination. There is one term called nitrogen balance. So do remember this term is very important for your grand viva. In your viva you have this term is most of the time asked, what is nitrogen balance, what is positive nitrogen, negative nitrogen balance. Proteins are the major source of nitrogen in the body. The urea excretion represents a net loss of nitrogen from the body, because the protein's end product of the metabolism of protein end product is urea. So it is the major nitrogenous, non-protein nitrogenous waste substance excreted from the body. So urea excretion represent the net loss of nitrogen. More urea excretion, more nitrogen excretion, it is said. And it should be compensated by dietary intake of the protein. Nitrogen balance means there is relationship between nitrogen intake and nitrogen excretion, and this balance, the intake and excretion, this balance is always in the equilibrium, it is always equivalent, and the intake is in the form of proteins and excretion is in the form of urea. So the intake and excretion is always in the equilibrium, and if it is in the equilibrium, it is said to be in nitrogen equilibrium or nitrogen balance. If it is affected, if intake is more than the output or vice versa, the conditions are there which are called as positive nitrogen balance and negative nitrogen balance. So anabolism is equal to catabolism. Depending on this, there are three conditions: nitrogen equilibrium, the intake is equal to output. The positive nitrogen balance, it is seen in growing children, pregnant women, and during recovery after serious illness. In this condition, as you can see, the nitrogen intake is more than the nitrogen excretion, because in this condition, in growing children, the protein requirement increases because of the increased protein, cell proliferation, cell formation, cell division, the tissue development. The protein's requirement is more. In pregnant woman, obvious, the fetus is growing, so protein requirement is higher. During serious illness, there is already during illness the protein damage is there. So during recovery, the protein intake increases than the excretion. The third condition is negative nitrogen balance. The opposite is the case. The intake is less than the nitrogen excretion. Okay. So like the condition as we discussed in the last lecture, starvation, there is no food. There is no food means no protein. So nitrogen intake is lowered but excretion continues. The Kwashiorkor marasmus, these are the protein energy malnutrition. There is deficiency of protein, there is deficiency of energy. So protein deficiency, that is lower intake of nitrogen in the form of protein but excretion continues. The illness, major injuries, trauma, also in diabetes mellitus and in advanced cancer. Because in this condition, the intake is affected or intake may be there but excretion is more due to protein breakdown. In these conditions, diabetes and advanced cancer, the protein breakdown increases. Now the fates of ammonia and carbon skeleton of amino acid. Every amino acid, as we know, has two functional groups, ammonia in the form of amino group and the carboxyl group. The first step in the conversion or the catabolism of amino acid is to undergo transamination followed by deamination, and after deamination the amino group is separated as ammonia. Ammonia is separated and remaining structure is called as a carbon skeleton. So what is the fate of this ammonia and carbon skeleton once amino acid undergoes catabolism? The products of carbon skeleton are mostly acetyl-CoA, pyruvate or some intermediate of TCA cycle like oxaloacetate, alpha-ketoglutarate and succinyl-CoA. So once ammonia is separated, the carbon skeleton, the remaining structure of amino acid, may be get converted to acetyl-CoA, enter TCA cycle, produce energy. It may be converted to pyruvate depending on the condition. The pyruvate may be converted to acetyl-CoA, enter TCA cycle, produce energy, or the pyruvate may undergo gluconeogenesis, converted to glucose, add to blood glucose or glucose supplied to brain, or it may form the intermediate of TCA cycle, like for example the amino acid aspartate when undergo transamination, the alpha keto acid, the carbon skeleton formed is oxaloacetate. It is one of the intermediate in the TCA cycle. Alpha ketoglutarate by the deamination or the transamination of glutamate, alpha ketoglutarate is formed. Also succinyl-CoA, which is a precursor for synthesis of heme, or it is also the intermediate of TCA cycle. So this is what happens to carbon skeleton of amino acids. They provide energy through TCA cycle because acetyl-CoA, one of the common collecting point of all macromolecules. Acetyl-CoA may be synthesized from carbon skeleton and it provides energy via TCA cycle. It can yield glucose by gluconeogenesis, as I told you, pyruvate and oxaloacetate. These are the non-carbohydrate precursor for the synthesis of glucose via gluconeogenesis. So it can yield glucose. It can form ketone bodies depending on the condition. The acetyl-CoA is synthesized first from the carbon skeleton, and this acetyl-CoA may be converted to ketone bodies. So as we know, the amino acids, it has an amino group. This amino group is separated by transamination and deamination in the form of ammonia, and this ammonia is toxic to the body. It mainly has the toxic effect on the brain. So this ammonia needs to be detoxified, and by the way of urea cycle, this ammonia is converted to urea, and this urea is comparatively non-toxic and soluble, so it can be easily excreted in the urine. The second functional group, or the whatever structure remains of the amino acid. So this is R is the side chain, the central carbon, the second functional group, and fourth is hydrogen. So remaining structure of these amino acid is called as carbon skeleton. So this carbon skeleton has various fates. It can be converted to acetyl-CoA. This acetyl-CoA may enter TCA cycle and produce energy, or it may take part in the ketogenesis, the synthesis of the ketone bodies, as it is precursor. Or this carbon skeleton can be converted to the alpha keto acids like pyruvate, oxaloacetate, alpha ketoglutarate and succinyl-CoA. and these are the intermediate of TCA cycle. They can complete the TCA cycle, or they may be utilized in the generation or formation of glucose via gluconeogenesis. The first, the any amino acid, when it is to undergo catabolism, there are some common reactions every amino acid undergoes. That include transamination followed by deamination and urea cycle. So these are the three common reactions every amino acid undergoes. So the first is transamination. The name itself suggests us trans means there is a transfer of amine group. There is a transfer of amino group. That's why the reaction is called as transamination. There is no separation of amino group as such. There is only transfer from one molecule, that is amino acid, from one amino acid to the second molecule to form the second amino acid. So it is a transfer of amino group from one amino acid to a keto acid to form a new amino acid and a new keto acid. So if you see this reaction, so this amino acid is supposed to undergo transamination. It has the amino group. This amino group from this first amino acid is donated to the keto acid. Okay, amino acid from amino acid is donated to keto acid. So this amino acid, by donating the amino group itself, get converted to a keto acid. So amino acid is converted to a keto acid. So difference between amino acid and a keto acid is, in amino acid the amino group is present, in keto acid amino group is absent. So first amino acid donate the amino group to keto acid, and amino acid itself get converted to a keto acid. And the keto acid, so the amino acid is converted to a keto acid, and the keto acid, by accepting the amino group, is converted to amino acid. So this is nothing but a transamination reaction. There is no net separation of amino group as ammonia in transamination, only transfer of amino group. And these reactions are catalyzed by the group of enzymes called as transaminases or aminotransferases. And this reaction or this enzyme requires a cofactor called pyridoxal phosphate. It is a cofactor derivative of vitamin pyridoxine. Okay, so transamination reaction catalyzed by the enzyme transaminases with the help of pyridoxal phosphate, the cofactor. There is only transfer of amino group from amino acid to keto acid, and due to this transfer the amino acid is converted to a keto acid, and the keto acid, by accepting the amino group, is converted to amino acid. So examples, the physiologically most important two reactions are there, two important transaminases are there. One is the alanine transaminase and second is the aspartate transaminase. So first reaction, alanine transaminase, or it is also called as SGPT, serum glutamate pyruvate transaminase, with the help of pyridoxal phosphate. This enzyme transfers the amino group from amino acid alanine to a keto acid alpha ketoglutarate. And when amino group from this alanine is liberated, alanine or the carbon skeleton of alanine is converted to alpha keto acid, that is pyruvate, and the alpha ketoglutarate, the keto acid, by accepting the amino group from alanine, the alpha ketoglutarate plus amino group is converted to the second amino acid, that is glutamate. So there is transfer of amino group from alanine to alpha ketoglutarate, and alanine is converted to pyruvate and alpha ketoglutarate is converted to glutamate. The second is AST, that is aspartate transaminase or serum glutamate oxaloacetate transaminase. This enzyme catalyzes the transfer of amino group from aspartate to alpha ketoglutarate. So the aspartate, by donating the amino group, it is converted to oxaloacetate, the keto acid, and the keto acid alpha ketoglutarate, by accepting the amino group, it is converted to the amino acid glutamate. Do remember the name of the enzymes given here is either ALT, that is alanine transaminase, depending on the substrate. The substrate is alanine for this type of transaminase, and in this reaction is AST, that is aspartate transaminase, because the substrate is aspartate. That's why the name aspartate transaminase. The other nomenclature is for ALT, it is SGPT. The full form is serum because the activity is measured in the serum in the clinical lab. It produces two products. By the action of this enzyme, it forms two products. One is G for glutamate, P for pyruvate. As you can see on the right hand side of the reaction, two products are glutamate and pyruvate. So serum, glutamate, pyruvate, transaminase, the action. Same way, serum, one product is glutamate, G for glutamate, second product is oxaloacetate, O for oxaloacetate, and the action, that is transaminase. The ratio of plasma levels of transaminases, that is SGPT or ALT to AST, is always 0.7 to 1.4, the normal ratio. It is altered in various abnormal conditions like drug hepatotoxicity, alcoholic hepatitis, chronic hepatitis, and decrease in acute viral hepatitis, because these two enzymes are principally present in the liver. Followed by liver, the heart is the principal site for the enzyme SGOT only. Okay, so any damage to liver affect this ratio, that may helpful in the diagnosis of these various conditions. Let me finish these characteristics and we'll stop for tomorrow, today's lecture. Yes? Yes, someone was saying something. So characteristics of transamination, it is universal and reversible. Universal in all organisms, transamination is followed by the same group of enzyme transaminases, and every amino acid undergoes transamination, and it is reversible. So as you can see in the previous slide, the transamination, the arrow is in both direction. The arrow is in both direction, that it is reversible. The first amino acid can be converted to second amino acid, or vice versa. Second amino acid can be converted back to first amino acid. So it is reversible. The cofactor or coenzyme PLP in the form of pyridoxal phosphate, active form of pyridoxine vitamin. Pyridoxal phosphate is required by all transaminases. No free ammonia is evolved. Do remember, the free ammonia is evolved or separated in the next step, that is deamination. There is only transfer of amino group. There is no removal of amino group. All amino acids undergo transamination except for lysine, threonine, and proline because of their complex structure. Then both catabolic and anabolic in nature, because on the left hand side the substrate is catabolized. The alanine is catabolized, broken down to its products, and on the right hand side some amino acid is synthesized. Some amino acid is synthesized, so it is anabolic in nature. So both catabolic and anabolic in nature. During protein biosynthesis, all required amino acids may not be readily available in the cells, because as we know, there are two types of amino acids, essential and non-essential. Non-essential amino acids are synthesized in the body, but essential amino acids may not be present. Essential amino acid we may take through diet, and non-essential amino acids are synthesized in the body. And one of the important ways of synthesis of non-essential amino acids from essential amino acids is the transamination. So essential amino acids can get converted to non-essential amino acids. For example, I'll give you of phenylalanine and tyrosine. Phenylalanine is essential amino acid, while tyrosine is non-essential amino acid. So tyrosine in our body can be synthesized from phenylalanine. Okay, so this is how the non-essential amino acids are synthesized. So conversion of essential to non-essential amino acids, various reaction is the transamination. So here we stop for today's lecture, and from tomorrow we will study about the important part, that is ammonia metabolism or the urea cycle. I hope you understood